Hello, everyone, and welcome to the 4th International APS Type 1 Symposium. My name is Jennifer Orange, and I am the Vice President of the APS Type 1 Foundation and the mother of an amazing girl with APS Type 1. With me to welcome you is Dr. Irene Lara-Corrales of SickKids Hospital in Toronto, our co-host today. Irene, would you like to say a few words?
Thank you, Jennifer. Good morning, good afternoon, or good evening, depending on where you are. It is a great honor for me to welcome you all on behalf of SickKids. I think it seems only yesterday that we hosted the first International Symposium here in Toronto, but it was 6 years ago, so it's quite a while now. I think it is great that despite our current reality, we're still able to gather virtually and carry out this wonderful event. And on behalf of SickKids, we are delighted to be able to support this event. Welcome, everybody.
Thank you so much, Irene, and thank you for helping me and the foundation get this entire movement started back in 2015. I also wanted to thank Carly Ramirez from the Dermatology Department for all of her support in getting this symposium ready. For our international attendees, in Canada, we begin important gatherings like this one by acknowledging the land and the people who have lived on this land. So to begin, this land where SickKids Hospital operates in the city of Toronto is the traditional territory of the Mississaugas of the Credit, the Anishinaabe, the Chippewa and the Haudenosaunee, and the Wendat peoples. And it is home today to many diverse First Nations, Inuit, and Métis people. We are grateful for the care that Indigenous peoples have taken for this land. And as a rare disease community, we know the important connection between the health of our environment and our physical and mental health. So as we meet virtually, I invite you today to take a moment to reflect on the history of the land and its peoples wherever you find yourself. Before we begin with our incredible speakers today, I just wanted to say a few thank yous. First, thank you to the board of the APS Type 1 Foundation, Sherry Seifert, David Seifert, Robin Finch, Heather Tallarico, and our president, Todd Tallarico. The work of the board, together with incredible support of Marilyn Sloan, has made this event possible. We would also like to thank the Foundation's Medical Advisory Board, Doctors Mark Anderson, Richard Aukus, Mihalis Leonakis, Dana Orange, and Maureen Sue. We are so fortunate to have your advice. Thank you. We would like to remind everyone about the Foundation's APS history. We are very near our goal of having 100 patients registered so that we can support our researchers as they work to discover better treatments and ultimately a cure for APS Type 1. More information on the registry can be found on our website at apstype1.org. I want to thank SickKids and the SickKids Foundation for their support at this event. And donors to Julia's Fund at the SickKids Foundation who are from all over the world. Your donations, together with the donors to the APS Type 1 Foundation, have made this event possible. Finally, to thank Dr. Tina Park and her associates at the Park Group for supporting this event. You have been such a pleasure to work with and have helped us reach so many new attendees, as we have now, I've heard 150 people from around the world attending. Thank you. Before we get to our incredible speakers, I want to remind everyone that nothing that is said today should be construed as medical advice. And now to our presentations. In the interest of time, I will not read the full bios of our speakers. They are extremely esteemed speakers, but you can find each of their full bios on the registration site. Each of our speakers will present for up to 30 minutes, followed by about 15 minutes of Q&A. Please type your questions into the Q&A chat, and we will try to get to as many as possible. So to begin, I'm very pleased to introduce Paul Bastard, who's joining us from Paris. Paul works with Necker Hospital and Rockefeller University. It is his first time speaking at our symposium, and we are so grateful to have you here. Welcome, Paul. Bienvenue.
Merci beaucoup. Thank you very much, Jennifer, and everyone in the APS-1 Foundation for the very kind invitation. I'm to be here. My only regret is that it's not in person, but hopefully soon it will be again. So as Jennifer said, my name is Paul. I work in Paris, and I'm a pediatrician and currently finishing PhD in investigation in the lab of Jean-Laurent Casanova at the Necker Hospital. And so today I will briefly introduce to you what our work has been during the last two years approximately, focused on COVID and neurons. But this work actually started many years ago, and several people in our group and in collaboration with many other groups have been working on the side before COVID. So the main topic today will be the autoantibodies against type 1 interferons in patients with APS-1. Before I discuss specifically APS-1 and to understand the context, I will try to introduce COVID-19, obviously, because it's one of the major subjects here, and also type 1 interferons, as some of you might not yet know what their function is, and also obviously what we found in regarding these autoantibodies in patients who do not have APS-1. So regarding COVID-19, we now have been living through this pandemic for almost 2 years, and most of you have seen these images of very severe lung affectation. This is a French patient who was hospitalized during the first wave at the very beginning of 2020. And although we know that there are very severe cases, these individuals with severe lung or what we call pneumonia are actually the rare kind, as most individuals have either asymptomatic infection, meaning no symptoms at all, they might not even know it, or very mild infection with flu-like symptoms. And in terms of risk factors of why certain individuals develop severe COVID-19, a few have been found very early on. The major risk factor at the epidemiological side is the age. This was very striking in the first months of the pandemic. And now we know that the risk of having COVID-19 pneumonia, severe, doubles every years. There's also a slightly higher risk for men, and there's a sort of between 5 and 10 other comorbidities like obesity that have been shown to underlie an increased risk of COVID-19, but they have all been described with some moderate risk. So here, the risk is less than twice the people who did not have the comorbidities. And even within a same group of individuals who have the same age, sex, and similar comorbidities like obesity or overweight, well, there remains huge differences between different individuals. Some might have very severe infection, while others, sometimes in the same family, might be completely asymptomatic. And we've heard in the newspapers that centenarians have actually handled the virus very well without having any symptoms. And sometimes there's actually been a few case reports of people who survived the Spanish flu in 1918 and who survived, now that they're over 100 years old, COVID-19. And on the other side of the spectrum, unfortunately, very rarely, young individuals have been found to have severe COVID-19 despite not having other factors to explain this severe disease. So the question that our lab and many other labs across the world has tried to tackle is, how do we explain such differences between individuals who at baseline seem to be similar? So the first attempt to answer this question, Jean-Laurent Casanova and Helen Su at the NIH teamed up with many other research groups, physicians all across the world to answer the question I posed just before. The idea was to try to recruit as many patients as possible with either very mild COVID, so no symptoms or flu-like symptoms, but no hospitalization, or individuals with very severe COVID-19. And the hypothesis was that the patients with severe COVID-19, that is written here, life-threatening, would have an underlying cause, either genetic, autoimmune, or other. And so this recruitment has gathered a lot of interest. And now within this Human Genetic Effort Consortium that is very international, there's more than 400 centers that participate. As of September 1st of this year, more than 10,000 subjects have been recruited. And for all these individuals, the goal is to sequence their exome or genome, which means looking at DNA in great detail, to try to see if they have genetic factors. So this will be the first sort of beginning of the introduction, the genetic factors, because it will lead afterwards to the much more frequent autoimmune factors. And so last year, while we had approximately 2,000 individuals recruited in our study, we focused our attention on one pathway. These are the type 1 interferons. So this, on the right, you can see a relatively complex scheme where there's actually 3 types of interferons, which are molecules that are very potent anti-pathogens, but they have very distinct functions. For example, type 1 interferons here are very important for the fight against viruses, and they've been described in the 1950s. And since then, it's been very clear that they're the most potent first line of defense against the virus. And when I say viruses, I mean any virus. On the other hand, type 2 interferons are very important for the fight against another type of pathogen, mycobacteria, which are, for example, tuberculosis. And type 3 interferons are known to be also important, again, to fight viruses, but most likely in other tissues than type 1 interferons. Now, we focused on type 1 interferons because we knew from our studies and studies of other groups that patients who have genetic deficiencies in any gene in this pathway could have a viral infection that would be more severe than patients who do not have genetic defects. What is interesting about this family is, although there's only one little cytokine, the one molecule, one type 1 interferon shown here, there's actually 17 different subtypes. And we'll go back to this a bit later, but it's very surprising because they all bind to the same receptors shown here, interferon receptor 1 and interferon receptor 2. And so these are produced minutes after viral infection and can induce an antiviral response very quickly. The whole signaling pathway that we see here actually takes place within minutes, and within a few hours, there's the production of a lot of genes that will participate in the fight against the virus. And so knowing this crucial role of type 1 interferons, we wondered if deficiencies in these type 1 interferons or in genes controlling their production or their response or their action could be the cause of severe COVID-19 in some patients. So based on this, we studied, this was last year, individuals with severe COVID-19. And here that was very striking because most of these patients were adults who had been hospitalized for the first time in the intensive care unit for COVID-19, but who had never suffered from viral infections before. Why is this surprising? Because in our mind, we thought that having a deficiency in type 1 interferons would be mostly found in children. In any case, in a small percentage, between 1% and 3% of these cases, we found genetic defects of genes controlling the type 1 interferon pathway that is shown in a little more detail here. And strikingly, we found deficiencies, a patient who had complete deficiencies in molecules that were crucial for the type 1 interferon response. For example, the gene here shown IRF7, which controls the production, or as I mentioned previously, IFNR1, one of the 2 receptors. And we know that if you lack this receptor, you're going to be completely unresponsive to type 1 interferons. And this is what is shown here on the right-hand side. This is an experiment that we did last year on the cells of one of the patients with suspected type 1 interferon deficiency because of a mutation in the receptor. And as you can see in the upper panel, when we stimulate the cells of a control, C3 here, with interferon alpha, which is one of the 17 type 1 interferons, or interferon beta, another one, there's a very clear response in terms of the dark line going to the right. On the other hand, when we look at the response in the cells from a patient with complete interferon deficiency, you see that there's no difference when we compare the unstimulated cells and the stimulated cells. Whereas when we stimulate these cells with another molecule, interferon gamma here, well, we see that the responds normally. So this means that this patient indeed had a defect in type 1 interferons because of this mutation in IFNAR1, the receptor of type 1. And so based on these findings, we found that another gene that's present on one of the sexual chromosomes, the X chromosome, could underlie severe COVID in more than 1% of men below 60. So following this, the action of genetic in a small proportion of patients, we wondered, well, could there be other mechanisms that could explain the deficiency of type 1 interferons? And so we wondered about the autoantibodies targeting these type 1 interferons, because these interferons are molecules that are circulating in the blood, so potentially they can be targeted by autoantibodies. And I'll get back to this in a second. So what are autoantibodies? Well, you all know it actually. The autoantibodies against interferons are like any other autoantibodies. They're antibodies produced, but instead of targeting a pathogen, target one molecule inside your cell. So for example, here, it's a little counterintuitive. The autoantibodies target its own immune system, here, interferon, for example. And so what has been known about these autoantibodies is that they can block completely the effect of these type 1 interferons. And here it's very surprising because they've actually been known since the 90s. They were found in patients in 1981 who were treated because of viral diseases, for example, such as hepatitis, with recombinant interferon. And that these unknown reasons developed autoantibodies that would block their treatment. They were also found a year later in women with lupus. These women with lupus are known to have a high interferon level, which is potentially one of the causes of their autoimmune disease, of some of their manifestations. And it was found then, and in many studies since, that some of these women could have autoantibodies that would block the interferons and that could make their lupic disease get a little better. But they were thought to be completely silent. And then I'll discuss this again in a minute, but in 2006, it was found that they were also present in all patients with APS-1. Nevertheless, since the 1980s, they were mostly thought to be completely silent. What we mean by this is that they were there present, but did not have an impact, and especially they were thought to have no impact on viral diseases. Nevertheless, when we go back again into the 1980s, a paper shown here by found that these antibodies led to a more severe varicella infection in a woman that was 64 and that had these autoantibodies before the infection. They were also found by Luigi Notarangelo's group at the NIH to underlie severe viral infections in patients with immune deficiency and autoimmunity. And then unfortunately, COVID-19 arrived in 2019, and in early 2020, we heard the case of 3 patients with APS-1 who were hospitalized in the intensive care unit because of critical COVID-19. And so at the time, we wondered if these autoantibodies could indeed sort of mimic the genetic defects and also underlie COVID-19. And so what we did in collaboration with the NIH in Bethesda and many other groups all over the world was to look in patients at the time who did not have APS-1, but who had severe COVID, for these autoantibodies against interferon alpha-2 and interferon omega. So one might wonder, why did we pick alpha-2 and omega while there are 17 type 1 interferons? Well, the reason is very simple, is that these were the ones that were found in 2006 to be present in patients with APS-1. Were used sort of as a screening test for APS-1 patients, almost as a diagnostic test before the genetic testing. And so when we screened these individuals with forms of COVID-19 pneumonia, we found a very surprising result, which was that a very large proportion, more than 10% of patients from the ICU had very high titers, as you can see here, antibodies blocking either interferon alpha-2 and or interferon omega. And initially, people asked us, well, could this be either irrelevant or caused by the infection? And so to try to answer these questions, we also recruited about 600 individuals with very mild infection or even asymptomatic, and found that none of them at the time had autoantibodies against these interferons. And finally, we looked into healthy individuals for which samples were collected before the pandemic, and individuals that were all below 65, and this is important, about 0.3% of them had these autoantibodies before COVID. And so this suggests that before COVID-19, some people have these autoantibodies against type 1 interferons, although they did not have APS-1, and apparently they did not suffer from other infections. What was also interesting is that for a few individuals, we found these autoantibodies to be present before infection, actually in all the individuals that were tested. And furthermore, at the time, we realized that most of the patients with autoantibodies were men, and that most of them were actually elderly. And here, potentially, this was a first explanation of why the two main risk factors that I told you about before were here, why men have more severe viral infections and why elderly people have more severe COVID-19 infections. And so after being replicated in several countries around the world, well, we teamed up with our collaborator at Rockefeller University, Charlie Rice, to try to demonstrate that these autoantibodies against interferons were indeed the cause of COVID-19 in these patients. What the team of Charlie Rice did was a very clean and almost self-explanatory experiment, although there's a lot of curves here. But basically, they took control cells that they infected with SARS-CoV-2, the virus causing COVID-19. And they've shown previously that when they infect these cells with SARS-CoV-2, there's a very high viral replication. And then what they did is treat the cells with interferon alpha-2, one of the 17 type 1 interferons. And when you treat the cells with interferon alpha-2, these are the two blue lines, you see that there's a complete blockade of the viral replication. So interferon alpha can block the virus and abolish viral replication. And this presence of interferon alpha-2 was added in presence of plasma from healthy controls. And so here, this is the proof that plasma from healthy controls, plasma is part of the blood where autoantibodies are present. Plasma from healthy controls has no effect on interferon alpha. On the other hand, when we take plasma from patients who have severe COVID-19 and in whom we found these autoantibodies that block interferon alpha, and we do the same experiment, you see here that there is a very high viral load and that the virus can replicate almost without any problem. And then they performed dilutions and found that even when you dilute the plasma 10,000 times, we can still see some virus replicating, meaning that these autoantibodies are actually present at very high titers and very potent. So based on these findings, this was last year in 2020, we also wondered if more patients with lower titers could also have COVID-19 because of these autoantibodies at more physiological titers. And I won't go into the technical details here, but what we did was set up a more sensitive assay, because what really matters about these autoantibodies is not only their presence, but if they can block the effect of interferon, meaning they can bind to it and block it. And so what we set up is a simple experiment in which we incubate plasma or serum from the blood of patients and interferons at different doses. And we were able to go at lower doses that are more what happens during COVID infection. And then we looked at the effect. And so if there are no autoantibodies in the patient's samples, well, the type 1 interferons, for example, interferon alpha, can induce a very high luciferase signal. On the other hand, if they are autoantibodies, shown here in this little sort of shape, that block the type 1 interferons that are shown in blue, well, there's absolutely no possibility of type 1 interferons inducing a signal because it cannot be functional. And we conclude that there are neutralizing autoantibodies. And here the neutralization is very crucial. And now a few months ago, we published again in a very large cohort of patients with severe forms of COVID that more than 10% had these autoantibodies, actually closer to 15% of the very severe cases. About 8% of the intermediate patients are those that were hospitalized but not in the ICU, and actually 1% of the controls. So this means that although these autoantibodies lead to a very high risk of severe COVID-19, it's not a 100% risk. And that there are individuals who have these autoantibodies and who do not develop severe COVID-19. And so finally, we wondered in the general uninfected population, what was the prevalence of these autoantibodies before COVID? Because we know that in APS-1, they're present in most individuals, but we do not know in the non-APS-1 population. And so for that, we recruited and tested about 34,000 people whose samples were collected mostly in blood banks before COVID-19 pandemic started. And we tested for these autoantibodies. And here it was very striking because we found that before 65 years old, in the younger individuals, the prevalence of the autoantibodies was relatively flat and quite rare, close to 0.2 to 0.5%. And here, around 65, 70 years old, there was a dramatic increase in the prevalence of these autoantibodies. And so potentially here, this is another explanation for why elderly individuals have a higher risk of having severe COVID-19 because they have more pre-existing autoantibodies against type 1 interferons. So now, how can we conclude about the genetic and the autoantibody causes of COVID-19 in the general population? Well, what we found in our studies is that about more than 14, closer to 15% of individuals who do not have APS-1 have, and who have severe COVID, have autoantibodies before COVID who block their type 1 interferons and block the antiviral defense, leading to a very high SARS-CoV-2 replication. As I mentioned previously, this is quite surprising because this is the immune system attacking another part of the immune system. but this can explain a part of the increased risk of COVID-19 in the elderly. And why are these findings important? Well, because looking for these autoantibodies is really easy. It's a test called the ELISA that takes a few hours, and that can be used at the early stage of COVID-19, ideally to give early treatment to these individuals. Now, our future plans for these autoantibodies in patients who do not have APS-1 are to trying to find the causes. Why do they appear? Why do they appear with age? And here, perhaps the fact that they're present in APS-1 will help us understand why, and I'll tell you a little bit more about it later. So now back to APS-1. As I mentioned previously, in 2006, it was described that most APS-1 patients have very high titers of autoantibodies here, as we measured, against interferon alpha-2 and interferon omega. Interestingly, most patients actually do not have autoantibodies against interferon beta or against the other interferon families. Here it's shown interferon lambda-1, the one that is mostly present in tissues. Why do we think that there's more autoantibodies against interferon alpha and omega? Well, because they're very closely related in their structure. They look very much alike, while on the interferon beta side, interferon beta has only 20% similarities with interferon alpha. And now, tested in our recent study with Michalis Leonakis, who is the next speaker, I believe, several patients with APS-1 who had COVID-19. And repeating most of the results of our colleagues in 2006, we tested for the 13 interferon alphas, for interferon beta, interferon omega, and then the two last interferons that I have not introduced yet, who are interferon kappa and interferon epsilon. And here we found that in all the patients that we tested, we found very high titers that are shown in red of the 13 interferon alphas, and in all patients, high titers of interferon omega. Most patients with APS-1, as shown previously, did not have the autoantibodies targeting interferon beta. Only one did. And rarely, we found one patient with autoantibodies against interferon epsilon. So what do interferon epsilon and interferon kappa do? We don't know exactly, but we know that they're for interferon epsilon in the female reproductive system and for interferon kappa mostly in the skin. But we do not know exactly if these autoantibodies against interferon epsilon could lead to any specific disease or any clinical manifestations. So based on our findings previously, on our knowledge that when you do not have enough interferon, or when you have autoantibodies that block interferons, and when you're infected with COVID, the risk of having a more severe disease is higher. We wondered, what about APS-1 patients? And again, through an international collaboration and supervised by Michalis, we tried to constitute a large cohort of patients with APS-1 who unfortunately had been infected with SARS-CoV-2. And so we heard about 22 patients from 7 countries, England, France, Italy, Russia, Scotland, Sweden, and the US, who had been infected with SARS-CoV-2. The patients who all had a confirmed diagnosis of APS-1, both at the clinical characteristics and at the genetic testing, were aged 8 to 48 years old. And in these patients, from these 22 patients, unfortunately, 86% had suffered from severe pneumonia, meaning lung infection with SARS-CoV-2 requiring hospitalization and oxygen supplementation. And 68% of them had actually required hospitalization in an intensive care unit. Although 2 had been completely asymptomatic and the infection had been actually diagnosed on a routine testing at school. So we reported these findings, and shortly after, we also heard of 4 patients with APS-1 from Germany who had been infected with SARS-CoV-2, but who did not develop any clinical manifestation, and the diagnosis was made retrospectively on the serology. So from these 2 studies, it looks like the patients with APS-1 who have autoantibodies against type 1 interferons indeed have a higher risk of having severe COVID-19, while this is all with incomplete penetrance. And so I think that Mihalis is going to show in a few minutes the treatments that were administered in the patients, so I won't go into details here, but I would just like to mention before finishing that potentially these autoantibodies can underlie other viral infections. They can underlie, in the recent study by Finnish and Sweden teams, perhaps VZV and HSV1 infection. And we also found that the autoantibodies against type 1 interferons could underlie adverse reaction to yellow fever vaccine, which is a live virus. So this is also a take-home message that when you have autoantibodies against type 1 interferons, you should avoid getting any yellow fever live attenuated vaccine. So overall, from patients who had severe COVID, we found that autoantibodies blocking type 1 interferons could lead to a severe COVID-19 infection, especially in elderly individuals. In the general uninfected population, the number of people who have these autoantibodies increase with age, and unfortunately, APS-1 patients are at high risk of having severe COVID, but not in all the cases. And as Mihalis will detail in a few minutes, vaccination and specific treatments can be done and can prevent severe disease. And finally, we conclude from these body of studies that type 1 interferons are essential for protective immunity against COVID-19. And as I mentioned previously, these findings have important implications for patients with APS-1 and for the general population, because these autoantibodies can be found early and can lead to specific treatments. And obviously, patients with autoantibodies, APS-1 or without APS-1, should get vaccinated as they are protected thanks to the vaccine. And perhaps finding these autoantibodies in APS-1 will help us understand the cause of these autoantibodies in patients who do not have APS-1, perhaps because of a link with the thymus. And with that, I'd like to thank you very much for your attention. Thank you very much for the invitation, and huge thanks to the physicians that participated in the study from all over the world, to the patients and their families, to you who accepted to give your samples and to help us understand better severe COVID-19 in APS-1, but much more broadly, actually. And I thank also Mihalis Dionakis, who's been, with whom it's been great to work with on this study and on other subjects, and Jean-Laurent Casanova and Laurent Abel, who lead our lab in Paris and New York, and then all our team in both Paris and New York, the autoantibody team, and finally, all the collaborators, physicians, scientists, the world who teamed up to understand APS-1 and severe COVID-19. And with that, I'm very happy to take any questions.
Thank you so much, Paul Bastard. Thank you for this important work and for this excellent talk. We're so grateful to have you here today. We have a number of questions in the chat, and I just, before we begin, wanted to especially thank you for calling out the patients who have provided samples and information data for your work. We know that our patients spend a lot of time and energy trying to support our researchers, and we appreciate those words of thanks. So let's dive into the questions. First, we have a question from a parent. Does a condition in which one has autoantibodies against interferon mean that they have a primary immune deficiency?
So now the answer would be yes. Having autoantibodies blocking type 1 interferons is very close. It's really a copy of having a deficiency in type 1 interferons, because the antibodies are able to block every type 1 interferon that is produced. And so the effects are the same. And it's a very specific type of primary immune deficiency that most likely for type 1 interferons affects viruses, and not all viruses, fortunately, but yes.
Thank you. And just to follow up from that, you mentioned the availability of the ELISA test, and perhaps Dr. Leonakis can speak to this too, but we're wondering as a patient group how available this test is.
So yeah, that's an important question too. So in France, it's available in about a dozen hospitals now that set up the ELISA test based on our protocol, and we're working with several private labs to have it available for the general public. We can provide the testing, and I know that Mihalis and the teams at the NIH can provide the testing for anyone as a research test. But I hope that with these findings, the test will be available for everyone in any lab across the street very soon. In France, it should take a few more months, but we're getting there.
Thank you. That's really helpful to know. And one other quick question before we dive into some other ones that I think require more detailed explanations. The 2 asymptomatic patients that you referenced in Germany, there's a question about whether they were very young.
So yeah, that's a very interesting observation and question. They were actually young, yes. The 4 patients in Germany and the 2 patients in our study were below 30. So there's potentially, an effect of age. This being said, we also had young patients who unfortunately had a severe infection. So being young probably doesn't abolish completely the risk, but the risk is most likely lower.
Okay, thank you. So we have a question from Dr. Deal, a great friend of ours. She's interested in the sexual dimorphism in severe cases. So you found that one of the genetic mutations in the interferon pathways was on the X chromosome. Are there genetic variants in the general population in this gene that might be protective for women or deleterious to men? Let's start there. Yeah,
that's a very that's a great question too. We focused on the X chromosome because obviously we knew of this high prevalence in men. So your question was our hypothesis initially. We have found this gene TLR7 that leads to a higher susceptibility. We have not found yet genes that are protective, but for sure there are variants that will be protective and others that will be leading to severe risks. And we're trying now to focus our studies on the X chromosome, on the Y chromosome, and on perhaps a sort of gene effect, because women who have 2 X chromosomes might have sort of a higher protection thanks to certain genes that are expressed twice, while in men, they're expressed only once. So it's a question. I don't have the exact answer, but I agree with the question.
Okay. And as a follow-up to that question, what is the relationship to sex hormones and COVID-19 infection and severity? It looks like women only become really susceptible well after the age of menopause.
So yeah, there has been several studies trying to understand the effects of sexual hormones. I don't think that any of these have pinpointed exactly why. Perhaps one of the explanations might be, I'm a little monothematic here, but the autoantibodies against interferons, because we have also found that although they're slightly higher in men, they're actually quite prevalent in elderly women. So I don't know why they arrive. And now we're trying to look at any effect, genetic, epigenetic, or hormonal, to explain why they arrive regardless of COVID infection. But it might be because of hormonal factors in men and women, actually.
Thank you. The next question is, how do these autoantibodies actually predispose to severe COVID-19? Is it the IL-1 pathway hyperactive in them?
So yeah, that's an important physiopathological question. So the way the autoantibodies work is that they're circulating in the blood, and whenever interferon, type 1 interferon is produced, they block it. And so when the virus arrives, our body produce type 1 interferons to block the virus. And so if there's enough type 1 interferon in the first hours of infection, well, to simplify, the virus is blocked. On the other hand, when you have the autoantibodies that counteract this type 1 interferon, well, the virus has no blockade and can replicate extensively and lead to a high viral load that might be symptomatic at the time or might lead to very high unnecessary immune response, for example, with the IL-1 pathway or the IL-6 pathway that creates a cytokine storm. So it's a sort of two-step model that we have. The first, because of the defective type 1 interferon, there's more virus. And then there's an overreaction of the immune system that really, and Mihalis will discuss it regarding the treatments, I'm sure, that would really be more deleterious than antiviral.
Thank you very much for that answer. The next question is, do you have any comments on APS-1, which is non-air, non-STAT2? And maybe you could explain what that means for our patients and families.
Sorry, APS-1 that is not
that is non
by
a mutation on the air gene. Yeah.
Right. So there are potentially other autoimmune genes that can cause autoimmunity. I'm actually, and I think that in these patients, a more broader genetic screening can explain the APS-1 or APS-1-like phenotype.
Thank you. I think Dr. Mihalis is also going to speak to this, but there's a question about vaccination. And are you still finding that vaccination is as effective in people with autoantibodies compared to the general population?
So yeah, I'm sure Mihalis will discuss it, but in our experience, we found that the vaccine response was very good in APS-1 patients, which was very reassuring for us. And we do not see differences in terms of response, meaning that the following 2 doses of vaccine, at least the serological response is good.
Thank you.
I don't want to, I don't know, give the information before Mihalis does.
I understand. Thank you. The next question is, why are there so many interferon alpha genes, but only one interferon beta or omega?
Well, that is a very interesting question. I would love to have the answer. I think this question has been on the 50s. I don't know. We're trying to work on this to understand why evolution has created so many interferons. One answer, an easy answer, is redundancy, because the more redundancy there is, the more possibilities of effectiveness there is. So people who would have antibodies against interferon omega only might have protection thanks to interferon alpha. But the why, unfortunately, we don't know yet.
Okay, thank you. I'm just going to pose 2 more questions as we have 2 minutes. And this question I love because it shows the generosity of our patients and families and their drive to help support all of your work. So this question is, do you still want samples from patients who have been vaccinated but have not gotten a booster yet? And if so, how do we provide a sample?
So thank you very much. We would be very happy. So we're looking at several things in patients who have been vaccinated, who have had COVID. We're looking at perhaps if there might be any change in the autoantibodies, and also at the vaccine response before and after the boost. So I think it really depends where you are around the world. If you are in the US, you can certainly ship samples to Dr. Ligonakis at the NIH. If you are in other countries in Europe or South America, you can ship to us in Paris. Maybe I'll write my email in the chat. And I'm very happy to discuss further about the logistics.
Thank you so much. And with our last minute, where do you see this research going in the future? What do you think the next steps are?
Yeah, that's a very important question. So here, what we're trying to do is we found that these autoantibodies could cause severe COVID. We're trying to see if they can be the cause of other viral infections. We've had a little glimpse at it. And now the big question is why they arrive in APS-1 in the general population. They might be closely related mechanisms, different ones, I don't know. And how can we prevent them specifically? Because they are obviously very aggressive immunosuppressive treatments that will work, and Mihalis will discuss it. But ideally, we would like to target these autoantibodies specifically without affecting the rest of the immune system. So that would be the ultimate goal, I think.
I like that goal. Thank you so much. And thank you, Paul Bastard, for this incredible presentation and for answering all of our questions. We're so grateful to have you here today.
Thank you very much. And thanks again. I mean, I really meant it. Thanks again to all the physicians and patients, because despite COVID-19 pandemic, everyone has been extremely helpful, and it's been a pleasure.
Wonderful. Thank you.